DNA sequencing is the process of determining the exact order of nucleotides in a DNA molecule. By identifying the sequence of these bases, scientists can study genes, genetic variation, and the organization of genomes. DNA sequencing is widely used in medicine, biology, and genetics for applications such as diagnosing genetic disorder, studying evolution, and developing personalized medicine. NGS, Next Generation Sequencing, refers to advanced DNA sequencing technologies that enable the rapid sequencing of large amounts of DNA or RNA. It also refers to the next advancement of sequencing technologies after traditional first-generation single sequencing. The common feature of short-read technologies is massive sequencing of short clonally amplified DNA molecules sequenced in parallel. NGS workflow includes library preparation, sequencing, and data analysis. Unlike traditional methods like single sequencing that use chain termination chemistry with modified nucleotides that terminate DNA synthesis at specific bases, NGS is high throughput, meaning it can sequence millions to billions of DNA fragments simultaneously in a single run. This makes it faster, more scalable, and cost-effective of analyzing entire genomes, transcriptomes, and targeted regions of DNA.
There are many key differences between single sequence and next generation sequence (NGS). Firstly, single sequence uses chain termination, while NGS involves parallel sequencing of millions of DNA fragments. Other than that, single method sequences one DNA fragments at a time, while NGS is massively parallel that sequences multiple fragments at one time. Furthermore, single sequences read length is long, up to 1000 BP per read, while NGS read length is short, typically 50 to 300 BP per read. Next, single process is slower because only processing few samples at a time, while NGS process is faster because it processes large amounts of data. After that, single sequence is expensive per base, especially for large-scale projects, but NGS is cost-effective for large-scale sequencing. Then, single sequence produces less data, while NGS generates massive amounts of data. Last but not least, single sequence has low error rate, while NGS has a slightly higher error rate but can be improved with coverage.
The advantages of NGS include its cost-effectiveness in screening large number of samples. NGS allows for the analysis of numerous samples at a significantly lower cost per base compared to single sequencing, making it ideal for large-scale studies and clinical applications that require high throughput. Another major of advantages is its higher sensitivity for detecting low-frequency variants. Unlike single sequencing, NGS can identify rare variants present in low proportion within a sample, which is particularly valuable in areas such as cancer research and rare diseases studies. Despite its benefits, NGS has certain limitations. One significant limitation is its restricted ability to detect certain types of variants. For example, NGS struggles with identifying large structural variants, copy number variants, CNVS, and epigenetic modification unless specialized protocols or sequencing approaches are applied. Additionally, most NGS platforms generate short read lengths, which can make it difficult to accurately assemble highly repetitive or complex regions of the genome. In such cases, long-read sequencing technology may be required to overcome these challenges.
The application of sequencing in genomic research. I have two examples: Illumina's NGS technology and Oxford Nanopore technology. Illumina's next generation sequencing NGS technology is powered by sequencing by synthesis or SBS, which use fluorescent lamp but reversible terminator nucleotide. The process begins with bridge amplification, where DNA libraries are clonally amplified on a sequencing flow cell using PCR. This step is automated and controlled by the sequencing instrument. During sequencing, a single fluorescently labeled DNTP is added to the DNA chain in each cycle by DNA polymerase. The system then captures this fluorescent signal, identifying the base. The reversible terminator and die are removed, allowing the next base to be added. This base-by-base sequencing ensures exceptional accuracy with an error rate as low as 0.1%, primarily due to substitution error. Illumina platform also support pair-end sequencing, where DNA is read from both ends. This enhances sequence accuracy, especially for applications like HLA typing by enabling tracing across polymorphic region. The MISEQ instrument, a common choice for HLA typing, works best with DNA fragment between 350 and 100 base pairs. ONT sequencing works by placing single-stranded DNA or RNA through a nanomer protein embedded in the flow cell membrane. DNA libraries are prepared by attaching adapters and a motoenzyme which cut the single strand through the nanopore. An applied ion current detects disruption caused by each nucleotide, translating this signal into sequence data. ONT ability to produce continuous long read depends on high molecular weight dating with standard reads ranging from 10 to 100 kilobase and ultra-long reads exceeding 100 kilobase. Despite its advantage, ONT raw read accuracy range from 87 to 98%, which limits its reliability for precise tasks like single nucleotide variant calling. Error often due to motoenzyme fluctuation or signal interference from neighboring base. However, as advanced in base calling algorithms are steadily improving accuracy. Beyond DNA sequencing, ONT can sequence RNA, including full-length native RNA, which avoid bias from PCR amplification. It can also detect modification in DNA and RNA by analyzing specific disruption in ion current. While ONT's error rate is higher than second-generation method, software correlation makes the data usable for many applications.